Hello everyone, and thank you all for listening to my talk. My name is Jolinda de Korne and I'm a PhD student at the Public Health Service of Amsterdam. And I want to tell you something about a project I did together with Odile Harrison from the University of Oxford, which was about the Neisseria gonorrhoeae accessory genome and its associations with the core genome. In the last decade, there has been a huge increase in the number of genomes available due to advanced sequencing techniques, and this allowed to do genome-wide comparison studies. And these studies have shown that the bacterial genome is not fixed, but rather consists of a flexible gene pool called the bacterial pangenome. And the pangenome consists of a core and an accessory genome, where the core genome is our genes, conserved genes, essential for survival and therefore found in all genomes of a particular bacterial species, whereas the accessory genome is found in only a subset of genomes of that particular species and offers flexibility to adapt to an environment or a host, and can also give advantage over other bacteria, which drives bacterial evolution as visualized here. There are closed and open pangenomes, and bacterial species with open pangenomes are able to adapt to multiple environments because each environment asks for a specific genome content, whereas species with a closed pangenome are more restricted and cannot adapt to that many environments. There are quite some genomes available for Neisseria gonorrhoeae, which are also quite well annotated. And in the PubMLST database, there is the core genome MLST scheme available, which defines the core genome of Neisseria gonorrhoeae. Also, there are some accessory elements known for gonorrhea, which mainly are plasmids. And one of the plasmids, the beta-lactamase plasmid, is able to transfer antimicrobial resistance. A current threat is that that Neisseria gonorrhoeae is developing resistance against third-spectrum cephalosporins, and this mechanism still contains an unknown factor X, as described by Unemo et al. And this gave rise to the idea that there might be an accessory gene, chromosomally encoded accessory gene, that plays a role in the resistance mechanism. However, the chromosomally encoded and accessory genome is not yet characterized. So that brought us to the project to characterize the Gonococcal accessory genome and determine its association with the core genome, which could inform us more about the population structure of the Gonococcus. First of all, I want to give some details about the methods we used for identification of the gonococcal accessory genome. We wanted to select a representative data set that represented all the isolates found in the PubMLST database. And that's why we selected 765 isolates, and we picked isolates based on the core genome MLST types in PubMLST, and from each MLST type we took some isolates, meaning that we had the full spectrum of core genome MLST groups in our data set. And here I want to show a minimum spanning tree of all isolates in the data set. And this tree is based on the core genome MLST scheme. The blue nodes represent isolates that we selected for our project. And as you can see, the blue nodes are randomly distributed throughout the tree, meaning that we did heterogeneous sampling, and that we have a data set that is representative for the isolates in PubMLST. So we worked with this set of isolates, and we used two different methods for identification of accessory genes. First of all, I used a PubMLST defined loci-based method by using the genome comparator plugin in PubMLST that can be found under the analysis tools in the database here. And this tool basically compares loci that you select within isolates that you select. So I selected all loci that are defined in PubMLST, and I wanted to compare them within the 765 isolates that I have in my data set. And the output shows whether or not a locus is present in an isolate. So I could select loci that were present in less than 95% of the isolates, meaning that those were potential accessory loci. Then I also used another method, a sequence-based method, because I thought maybe there are genes not identified yet in the PubMLST database, which we might miss by using a loci-based method. So I also used bioinformatic tools, Rory and Pirate, which are quite known for pangenome analysis. And these tools basically take annotated genomes, cluster coding sequences based on similarity, and the output file shows a list of genes that were found in the database, in the data set that you give to the tool, and it shows the prevalence of that certain gene in the data set. So also here I could select genes that were present in less than 95% of the isolates and that were potentially accessory genes. And I compared both methods, and I came up with a list of accessory genes that were together the accessory genome of gonorrhea. One very nice feature of Rory also is that it gives pangenome plots, which I'm showing here as the first result. So the plots show the number of genes versus the number of genomes added to the or given to the tool, and the straight line shows the number of new or conserved genes, conserved in the right plot, and the dashed line shows the number of unique genes or total genes. And as you can see, the number of new genes identified already satirizes when adding less than 50 genomes, whereas the number of unique genes kept increasing, meaning that the variation within a gene increased when adding genomes. However, the number of genes satirized already quite early. And we could conclude from that we are actually looking at a closed pangenome. And another thing that we could conclude from this is that our data set of 765 isolates is enough to identify the complete accessory genome. So after identifying all the genes in the accessory genome, I wanted to functionally annotate the genes and see what the accessory genome consisted of. So what was interesting to see is that almost half of the accessory genome consisted of already known mobile genetic elements, such as the GGI and the plasmids. And another interesting finding was that also a major part of the accessory genome consisted of phage-associated proteins and hypothetical proteins. And of course, hypothetical proteins are difficult to interpret because we don't know what they do or what they mean. And then we still had some genes with other functions that were also part of the accessory genome. Now we characterized the accessory genome, we were able to create an accessory genome MLST scheme in PubMLST based on all the genes that we identified to be accessory. We made this scheme, and based on this scheme, we made a minimum spanning tree, including all isolates available in PubMLST. And the colors here in this tree correspond to the core genome MLST group that isolate belongs to. And as you can see, the colors very nicely cluster together, meaning that the accessory genome is strongly associated to the core genome, which is also consistent with the clonality that we see in the Gonococcal genome, and which is now also found for the accessory genome. So the take-home messages from these results are that the Gonococcal accessory genome, first of all, is limited, and if we look at the lifestyle of gonorrhea, we could say that this is due to its obligate pathogenic and host-restricted lifestyle, so it doesn't need a huge accessory genome. Secondly, it consists mainly of mobile genetic elements, which we already knew, but also prophages and hypothetical proteins are a major part of the accessory genome. And lastly, it's also associated with the gonococcal core genome, which confirms the clonality of the gonococcal population structure. So I want to thank Odile for the great supervision that she gave me during this project. We wanted actually to meet in person, but all our meetings were online so far. But many thanks for the opportunity to do this together. And I want to thank also Martin, Sylvia, Ali, and Henry, my other supervisors. Thank you all for listening, and I would be very happy to answer your questions.